السلام عليكم طلاب، ان شاء الله مثل ما وعدتكم راح ان شاء الله اسجل هذه السلايدات الاولى. خاف طالب ما لحق خلينا نقول يترجم السلايدات الاولى، واضح؟ وان شاء الله قلنا اليوم اللي اخذناها الفيديوهات وجروبات امس ايضا راح ارفعها لكم ان شاء الله كلها في فيديو واحد. فتطرقنا الى البوليمريز تشين رياكشن ومختصر البي سي ار. شنو قلنا؟ تم اخذه من الاحرف الاولى الكابيتال، مو تمام؟ كابيتال بي وكابيتال سي وكابيتال ار. وقلنا بوليمريز تشين رياكشن. المقدمة البسيطة انه هو originally developed، طور متى؟ 1983 في 1983 by the American biochemist من طلاب كاري مولس. واضح؟ هذه طلاب صورة العالم اللي إلا الفضل جدا بكل التطور طلاب اللي جاي نشهده وبكل النواحي، واضح؟ ومختلف العلوم الأخرى. فبما انه مادتنا موليكولار فمية بالمية البي سي ار is used in molecular biology to what? To make many copies of، هذه خلينا نتركها، of what small section of DNA or a gene. واضح؟ إذن يستخدم لتكوين العديد من النسخ من قطع صغيرة من ال DNA أو الجين. زين. amplify طلاب، كلمة amplify يعني تضخيم، واضح؟ using PCR it is possible to what طلاب؟ نقدر نولد 1000 to مليون، آلاف إلى ملايين النسخ of what particular section of DNA from a very small amount of DNA. إذا تذكرون أكدنا على هذه العملية، قلنا إذا نسأل السؤال هل عملية البي سي آر تتم عن طريق استنساخ أو تضخيم the whole genome؟ قلنا أنتم بكل بساطة شنو راح تجاوبون؟ أكيد false، مو تمام؟ هو فقط particular section، مو تمام؟ وهذه طلاب انه polymerase تعني replication of the target sequence. شنو يعني؟ تضاعف التسلسل الهدف اللي انت تريده. و chain reaction طلاب انه هذا السيكونس راح يكون له repeatedly replicated. شنو يعني؟ راح يكرر بشكل دوري، واضح؟ يعني التضاعف. فطلاب لو اتينا الى البي سي ار is means to amplify a particular piece of DNA، مو تمام؟ شنو قلنا؟ انه هو يضخم فقط بيس قطعة وبارتيكولر محددة او معينة من ال DNA. ولو اتينا الى تعريف الامبليفاي يعني كلمة التضخيم، شنو نقصد بيها؟ making numerous copies of a segment of DNA، يعني تكوين نسخ numerous طلاب عديدة او هائلة من قطعة هذه segment قطعة ال DNA. PCR can make، شوفوا طلاب، مو فقط 1000 to مليون، حتى billions of copy، واضح؟ of a target sequence of DNA و in a short period of time، وبفترة زمنية قصيرة. هذا طلاب الصورة على بساطتها. تلاحظون هذا اللي باللون الأزرق؟ هذا طبعا شنو هو؟ DNA، مو تمام؟ تلاحظوا لي اكو فد هذا خلينا نقول شريط أو جزء باللون الأحمر، خلنا نعتبره طلاب هو هذا التارجت سيكونس، يعني الآن أريده وبعد ما خرج طلاب من جهاز الثرموسايكولر تلاحظون أي عدد هو صار له كوبي، نسخ عديدة، اللون الأحمر، مو تمام؟ اللي هو هذا التارجت سيكونس اللي أقصد به، يعني التسلسل الهدف من ال DNA قليل أو نسخة من التارجت سيكونس DNA. وتلاحظون بعد ما كمل جهاز الثرموسايكولر أصبح عندي نسخ عديدة. اتفقنا طلاب مهما اختلفت قلنا التصاميم، شنو طلاب؟ المبدأ واحد، مو تمام؟ وشنو سمينا الجهاز؟ ثرموسايكولر. وعرفناه أيضا بشكل بسيط، شنو قلنا؟ is a machine that heat and cool down، مو تمام؟ إنه هي آلة أو ماكينة بها حرارة، مو تمام؟ وكول داون وبرودة. وهسه إن شاء الله واليوم أعتقد أيضا صارت عندكم الصورة أوضح العملية من تطرقنا إلى الستيج، مو تمام؟ المراحل. وإن أيضا short period of time بفترة زمنية. خلينا نقول يعني العفو نقطة واحدة من هذه ما توفرت ما أقدر أسوي بي سي آر. فنمبر 1 the DNA template، شنو قلنا؟ قالب ال DNA. زين. ونمبر 2 primers، تقدر طلاب تعرف لي يا short، هذه إذا صعبوا عليك كلمة stretches اشخطها واكتب PIECE، واضح؟ S شو تصير؟ pieces، تمام؟ قلنا شنو؟ قطعة يعني، فهي قطع قصيرة short من ال DNA that initiate، هذه مرت علينا، مو تمام؟ التي تبدأ the PCR reaction، تفاعل ال PCR. design to what؟ هذه طلاب G silent، يعني تصمم to bind to either side، شنو؟ ترتبط من أحد الجوانب of the section of DNA you want to copy للقطعة اللي أنت تريد تضخمها. نمبر 3 DNA nucleotide bases. شنو قلنا؟ القواعد نيوكليوتايد also known as DNTBS، وعرفنا هذا المصطلح، واللي اتبع الطريقة 100% طلاب راح يكون عنده فرق وسهلة وحتى بالإملاء ما يخطأ، اللي هي طريقة التجزئة. مو تمام؟ فهذه ديوكسي رايبونيوكليوتايد ترايفوسفيت. والدي ان اي بيزز، هذه الاي ترمز الى الادينين، والسي سيتوسين، والجي جوانين، والتي ثيامين. هذه طلاب شو اعتبرها البيلدينج بلوكس، يعني الحجر الاساس للدي ان اي are needed to construct the DNA of العفو the new strand of DNA. احتاجها انا بالبناء، صح لو لا؟ انا مو راح اسوي نسخ، ف100% لازم شنو عندي طلاب؟ لازم عندي خلينا نقول الحجر الاساس اللي ابني بيه. مو تمام؟ ورقم 4 tag polymerase enzyme to add in the new DNA bases. شنو يعني؟ من اللي راح يبني باستخدام القواعد tag polymerase enzyme. واضح طلاب؟ هسه ان شاء الله مثل ما قلنا هذا السلايد ما مطالبين به. وهذا طلاب البفر رقم 5 اللي هو to ensure the right condition. شنو يعني؟ حتى يأكد انه هذه الظروف كلها طلاب ملائمة وتسير بالاتجاه الصحيح. واضح؟ هذه نفس الصور شنو نحتاج احنا للبي سي ار، وهذه ايضا نفس المكونات الخمسة بشكل مختصر طلاب. هذه الستيجات اللي موجودة طلاب بالفيديو السابق، ان شاء الله انا راح انتقل الى الفيديو. هذا ان شاء الله الفيديو الاول طلاب.
A sample of chromosomal DNA, also called the genomic DNA, can be used as the starting material for the polymerase chain reaction, also known as PCR. With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest or target DNA is indicated as red. In addition to the target DNA, a PCR reaction contains several other ingredients. These include free nucleotides, DNA primers, and the enzyme Taq polymerase. The primers are typically about 20 nucleotides long and are complementary in the sequence to the ends of the target DNA. Taq DNA polymerase is delivered from hot springs bacteria and can tolerate intense heat for the PCR reaction. A PCR reaction lasts several hours and typically consists of 20 to 30 repeating cycles. A cycle begins by heating the reaction mixture to 95 degrees Celsius. The heat denatures the DNA strands, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees so that the primers can form hydrogen bonds or anneal with their complementary sequence in the target DNA. Note that the primers and the target DNA follow base pairing rules. An adenine pairs with the thymine and a cytosine pairs with the guanine. In the next phase, the temperature is raised to 72 degrees Celsius. A Taq polymerase functions optimally at the temperature and begins to polymerize, adding nucleotides to the 3' ends of each primers attached to the DNA strand. After one complete cycle, there are two double-stranded copies of the target DNA formed. The PCR reaction mixture contains mainly copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA produced. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence, and note that only 2 of the double-stranded copies consist of just the target fragment, and the others also including flanking DNA regions. As the number of cycles increases, the products consist of greater proportion of the fragments with just the target DNA increases. After 4 cycles, half of the fragment consists of just target DNA and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of 25 cycle, there are more than 33 million copies of this double-stranded target region found.
Polymerase chain reaction, or PCR, uses repeated cycles of heating and cooling to make many copies of a specific region of DNA. First, the temperature is raised to near boiling, causing the double-stranded DNA to separate or denature into single strands. When the temperature is decreased, short DNA sequences known as primers bind or anneal to complementary matches on the target DNA sequence. The primers bracket the target sequence to be copied. At a slightly higher temperature, the enzyme Taq polymerase, shown here in blue, binds to the primed sequences and adds nucleotides to extend the second strand. This completes the first cycle. In subsequent cycles, The process of denaturing, annealing, and extending are repeated to make additional DNA copies. After 3 cycles, the target sequence defined by the primers begins to accumulate. After 30 cycles, as many as 1 billion copies of the target sequence are produced from a single starting molecule.
We live in a moment where genetics is helping us to understand more and more of the world around us, from untangling evolutionary histories to more precisely diagnosing and treating disease. But to do all of this, we need to be able to find and examine specific pieces of DNA. One of the fundamental tools that we use to do this is called PCR, or polymerase chain reaction. I'm Dr. Alex Danis with Mini PCR Bio, and today we're going to explore this important laboratory technique. PCR begins with a DNA sample, and DNA samples can be complex. Imagine the human genome with 3.2 billion base pairs. Trying to identify just one genetic sequence, which may be only a few hundred bases long, out of the entire genome of billions of bases, can be like looking for a needle in a haystack. PCR is a tool that finds that needle and then makes copies of it over and over until you are left with a pile of needles so large it's hard to find the original haystack. So how do we find our genetic needles in our haystack of DNA? A lot of it depends on the fact that our DNA is composed of 2 complementary strands that form a double helix. An A on one strand will always bind to a T on the other, and a C on one strand will always bind to a G on the other. This means that if you know the sequence of one strand, you always know how to replicate the other half. PCR is composed of 3 main steps: denaturation, which separates the 2 complementary strands of DNA; annealing, which specifies the region of DNA to be copied; and extension, which copies the DNA. These 3 steps will then be repeated over and over again. Now let's take a deeper look into how all of this works. The first step, called denaturation, involves separating the 2 strands of a starting DNA sample apart. This starting DNA sample is referred to as our template DNA. To do this, we heat the DNA up to about 95 degrees Celsius, almost reaching the boiling point of water. In just a few seconds, the high temperature causes hydrogen bonds between the 2 strands of DNA to break. After an initial longer denaturation to start off the PCR, each cycle requires a shorter denaturation, which can be as little as 10 seconds, but is often between 30 seconds and 1 minute. In the second step of PCR, we specify the region of DNA to be amplified. We do this using short pieces of DNA called primers. Each reaction will have 2 types of primers, forward and reverse. The forward primer will match a sequence on one strand of DNA at the beginning of the region we want to amplify. The reverse primer will match the end of the region on the other strand. Primers are typically around 20 bases long. When we cool our reaction down, the primers will anneal or stick to their complementary regions on the template DNA. Lowering the temperature allows hydrogen bonds to form between the primers and the template at areas where the sequence is complementary. We add lots of extra primers to increase the chances that they stick to the DNA rather than the DNA strands annealing back to each other. Annealing temperatures are typically between 50 to 65 degrees Celsius, but the exact temperature you use in your reaction depends on the specific length and sequence of the primers themselves. Our reaction may be allowed to anneal for as short as 5 seconds or as long as 30 seconds. Next, we heat the reaction back up again to extend or copy these new double-stranded areas where primer and template DNA have annealed. For this, we use an enzyme called a DNA polymerase, the P of PCR. The polymerase will find the ends of the short double-stranded regions of DNA where the primers have bound. It then moves along the DNA, base by base in the 5' to 3' direction, and adds the correct complementary DNA nucleotides as it goes. If it finds a C on the template strand, it adds a G. If it finds an A, it adds a T, and so on, creating a complementary strand of DNA. Our extension step can vary in time. When our target is long, we can extend this step to a few minutes long to give the polymerase time to copy the entire region. If we are copying very short regions, it may only need a few seconds. Extension typically happens around 72 degrees Celsius, the optimal working temperature of the polymerase. Of course, 72 degrees Celsius is hot. Our own body temperature is around 37 degrees Celsius, so if we used a typical human or bacterial polymerase, it would likely denature and stop working at these high temperatures. So instead, we use a special polymerase called Taq polymerase. This enzyme was originally found in thermophilic bacteria living in the hot springs of Yellowstone National Park and remains stable in our reaction even through multiple rounds of heating and cooling. All of these steps happen in a machine called a thermal cycler. Here, we are using a mini PCR machine from Mini PCR Bio. We place our DNA along with all the ingredients needed for the reaction into tubes and then place them inside. The thermal cycler will heat it up and cool it down just as we tell it to. After one cycle of these three steps, denature, anneal, and extend, we have turned our original one piece of target DNA into two. But we don't want to stop there. We repeat this cycle over and over again, now turning our 2 into 4, then 4 into 8, 8 into 16, and so on. The process creates new identical copies of DNA. We are exponentially amplifying our DNA. This chain of copying explains why it's called polymerase chain reaction. Most PCRs will run for about 30 cycles, which should give us about 1 billion copies of our original target sequence for every one copy we started with. For all of this copying to work, we need the exact right ingredients. So let's take a closer look at exactly what we put into each tube. First, we need our DNA template. Remember that this template is often a part of a complex DNA sample. Depending on the experiment, this DNA could come from almost anywhere, including a human blood sample, a water sample, a food sample, or even a bacterial swab. We also need our DNA primers, which specify which area of DNA we want to amplify. The DNA sample and the primers are unique to each experiment, but the rest of the reagents are the same in almost every PCR. We need the Taq polymerase, which will do the copying. We also need lots of spare nucleotides or dNTPs, the building blocks of DNA. These are the molecules the polymerase will add to the growing DNA strand. Finally, all this happens in a special buffer which contains salts and ions that help keep the polymerase functioning. What we do with our new DNA copies after our PCR finishes depends on our experiment. We may insert it into a plasmid so that a bacteria will produce a certain protein that we are interested in. We may use it to do DNA sequencing so we can learn the exact sequence of nucleotides in our DNA sample. Often, we will run it on an electrophoresis gel so that we can visualize the size of our DNA fragment or confirm that the PCR worked as we expected. This ability to make billions of copies of a specific DNA sequence has made PCR a staple of the molecular biology lab for decades and allows everyone from students to scientists to investigate DNA in the world around them. Mini PCR thermal cyclers continue to make PCR accessible to users around the globe, from classrooms to remote field outposts, and even up in space. To learn more about mini PCR thermal cyclers and learning labs that you can use to implement PCR and molecular biology techniques in your classroom, you can visit minipcr.com.